Thank you for the opportunity to talk to this group, which is a group I'm very fond of. So I think, is there a problem? Oh, I think quite some of you know, certainly people who know me for some years know that about 7 or 8 years ago, I took a bit of a strange left turn in my research. Up until then, I've been studying plant evolution, plant development, genetics. And then I started developing this interest and passion for the study of the origin of life. Initially, I thought this was going to be sort of just like a little digression, but it has taken over my life. And that is all I'm doing now, is studying the origin and early evolution of life. And in some sense, what I want to do today is talk you through what we've been doing and how we're approaching this problem, and why I got involved in the first place as somebody who views themselves first and foremost as an evolutionary biologist. So I like to start when I can with our good old friend Charles Darwin. And this is a very famous quote from the very end of The Origin of Species, which I think expresses the motivations many of us share for understanding, diverse life in general. It is interesting to contemplate an entangled black bank clothed with many plants of many kinds, with birds singing on the bushes, with various insects flitting about, and with worms crawling through the damp earth, and to reflect that these elaborately constructed forms, so different from each other and dependent on each other in so complex a manner, have all been produced by laws acting around us. So, of course, Darwin's agenda here was to remind us how awed we should be at the stuff around us, at biological diversity, at its seeming complexity, to use the word elaborately constructed forms, and we should seek explanations for this. And of course, his work laid the foundations for a big part of that, which is explaining how it is that once you have some kind of biological organism, complexity can accumulate through a process of natural selection. So what Darwin developed and what was built upon Darwin was this notion that complexity can increase over time, and complexity by which I mean these elaborately constructed forms. And thank you. Feel free anytime to do anything up there. And that it does that because mutation generates variation, that some of those variants might have higher complexity, but if they're selected for, then they can go to fixation. So there is a mechanism that Darwin provided that can explain how elaborate forms can arise once we have Darwinian evolution, as it came to be called. Of course, the problem that intrigued me is, okay, but in order for this all to work, we need to have Darwinian evolution. What is Darwinian evolution? Well, Darwinian evolution is evolution where we have organisms or cells that have some kind of inheritance, which is based on genetic machinery. This genetic machinery is tremendously complicated. And so we have this sort of the ultimate chicken and egg problem at the origin of life is in order to get complexity, we need evolution. Seemingly, in order to get adaptive evolution, we need complexity. So how do we resolve this? So, of course, I was interested, I think many of us in this room were interested in this question. And I came to feel that there were ways of thinking about this problem that were not being taken, partly because most of the people studying the origin of life are coming at it from a chemistry, physics, astronomy, geoscience end of things, and relatively few from the perspective of somebody trained in ecology and evolution. And so over time, this view sort of matured into this hypothesis, which is basically what I would say my lab is now testing. And this hypothesis is that at the beginning, there was a mode of adaptive change that looked much more like ecology, looked much more like ecological succession. And over time, that mechanism gave rise to the components needed for familiar Darwinian evolution. So in the beginning, there was change like succession. For those of you who don't know what succession is, this is where you have ecological communities that change over time as species disperse in, establish, maybe displace existing species. And there are some well-known patterns in how succession happens in natural communities. So in order to address this question of test this hypothesis, we first have to kind of come up with a way to structure the analysis so that we can make some formal equivalences. In order to say succession-like change and Darwinian-like change are continuous, we need to kind of define terms a little bit. And the way we have done that is by coming up with the notion, unfortunately partly cut off there, of an autocatalytic chemical ecosystem process. So I'm going to be presenting the view that both Darwinian evolution, familiar evolution, ecological succession, and a lot of other kinds of biological change can be described in this framework of autocatalytic. Come on, guys.
I'm so sorry.
No worries. No. A valiant effort. Let me see if I can get to the right screen for you. What did you? Let's move this like down here, maybe. Okay. So the idea. So this, they can be described in this general framework. So what are the units there? Well, these dotted lines here, these are ecosystems. They may or may not be bounded. An example of an ecosystem would be like a patch of sandstone in a forest in the landscape, or maybe it would be a mountaintop. But it could be bounded, and a cell, as you will see, is in this notion an ecosystem. Now, within these ecosystems, what makes them autocatalytic chemical ecosystems is that within them, they have these things called autocatalytic chemical cycles. Autocatalytic cycles, I'll unpack them in a minute, but you can think of them as little self-amplifying, self-propagating subsystems that interact with one another ecologically. So they're represented in that little central one there by little Pac-Man. Each of those is an autocatalytic cycle. And if you look closely, you can see that those autocatalytic cycles, some of them are interacting negatively with each other. One of the hammer and is knocking on the others. Some of them are positive, they're shaking hands. And then meanwhile, there are resources, little dots that are being exchanged among them or being provided to them as food or generated as waste. So that's what one ecosystem is. But in order to actually make sense of this, you'll see we can't just look at a single ecosystem. We have to look at a set of ecosystems that are connected to some degree. And we're going to call that a meta-ecosystem. So I might use the acronyms ACE for an ecosystem, an autocatalytic chemical ecosystem, and an ACME for an autocatalytic meta-ecosystem. So I'm going to argue and present that both succession and evolution can be described using the same kind of framework. But let's start with the autocatalytic cycles, and let's start with ecology, regular old ecology. Well, within an ecological community, the most obvious autocatalytic cycles are the life cycles of organisms. So if we have an environment that has plenty of all the food needed, all the food that's necessary, nothing will happen unless you introduce one individual that can trigger a self-propagating cycle. So let's say you introduce an adult, this adult, if I can find my little thing here, which I can't see. There it is. So this adult can give rise to multiple eggs, and that division process is called a branching reaction. It requires food, it might generate waste. And of course, because there are more than one egg produced, as you go around this cycle, the count of eggs and adults can increase indefinitely until limits are placed on it by the environment. You can draw the same thing in a slightly different way. If you have one adult that survives, lays one egg, survives to lay additional egg. But the basic idea here is that an organism, it's a set of chemicals. That set of chemicals goes through some kind of life cycle where it uses resources from the environment to make more of itself. And the stoichiometry, the count of individuals, keeps increasing. Now, those are not the only autocatalytic cycles in an ecosystem, because pairs of species and sets of species can interact in various different ways. And those can generate higher level autocatalytic systems. So it's a very simple case. Imagine you have two obligately mutualistic species, where one produces as waste a food that the other one needs, and vice versa. They form a higher level autocatalytic cycle, where both of them need to be introduced, but once they're both introduced, they can all members of that composite cycle can increase in number. So that's in a regular ecology. Now, within those organisms, within the cells of those organisms, there are also autocatalytic cycles. So if you go into metabolism, and you ask a biochemist, are there autocatalytic cycles in our cells or in the cells of other living organisms? They'll say, yes, there are a few. There are a few named autocatalytic cycles. So for example, the most famous one in origin life research is one called the reductive TCA, which is a cycle that feeds on carbon dioxide and reducing power. And every time you go around the cycle, the citrate splits into two, so you increase count as you go around and around. Now that's a named cycle in metabolism. It turns out there are many more. Jen Peng, who was a postdoc in my lab and is now a postdoc in bacteriology, he developed an algorithm for searching in biochemical networks. And it turns out there are uncountably many autocatalytic cycles hidden within conventional metabolic networks. And there's one shown there where the blue are the food that cycle needs, and the black are the waste, and it's a bit more complicated, but it still computationally can be proven to be autocatalytic, in that the stoichiometry, the counts increase as you go around the cycle. So those are some purely metabolic cycles, but if you think about it, within a cell, there are a lot of catalysts. If a catalyst, like a protein, catalyzes a reaction that promotes its own production, so here we have an enzyme Z catalyzing the reaction to make Y that is needed ultimately to make Z. Well, that is an autocatalytic cycle as well. If you redraw this, you can actually show that there's a substrate catalyst complex divides into the catalyst and the other path generating Y. So every reaction where a catalyst, like a protein, does something that benefits the production of that protein and all proteins, that's also another autocatalytic cycle. So there are hundreds of those in our cells. And then another important kind of cycle, a special case of catalysis, which is the catalytic cycle that involves the replication of genes or of DNA or RNA. So in this case, we have an autocatalytic cycle where we have a strand of DNA, feeds on DNTPs to make a double strand, to make a second strand, which splits. One of them can go directly, one of them is the catalytic strand, it's just recreated. The other one can be converted back to itself via the complementary sequence. So really, we have, in fact, two obligately mutualistic autocatalytic cycles here. But again, it's autocatalytic cycles. So cells are full of autocatalytic cycles. And when you study them computationally or theoretically, you can show that in the same way that pairs of species in an ecosystem can show diverse ecological interactions with one another, the same is exactly true for chemical autocatalytic cycles. You can show that two cycles can compete if they need the same food or they generate the same waste. They can be mutualistic, where one produces something the other one needs. They can be predators, where one eats the other, one eats members of the other. And they can be mutually inhibitory in various ways. And it turns out mathematically, these behave just like in the ecological case. So again, Jen in an earlier paper showed that all of these scenarios lead to very familiar ecological dynamics, but in chemistry. So for example, just predation, if you have a predator and a prey cycle, you can have either the predator can go extinct, or they can coexist, or they can show oscillations, either damped or stable, just like in regular ecology. So the upshot of this is that when we see a cell, we should see a subcellular chemical ecosystem that is highly analogous, if not identical, to the ecosystem that we might see if we go out into a natural environment. Within those, there are many autocatalytic cycles, sets of chemicals that collectively promote their own production, and they're interacting with one another in various ways. Sometimes they're helping each other, sometimes they're competing, inhibiting, and so on. So there's this equivalence between organisms and cells and ecosystems. which is going to provide it's sort of a basis for what I'm going to go on to now. So I'll pause just in case there are burning questions. Okay, that's the basic equivalence, but I haven't mentioned change, haven't mentioned everything. So let's start with Darwinian evolution. We're familiar with that. So what is Darwinian evolution? Well, conventionally, a traditional definition of evolution is a change in the frequency of an allele in a population over time. Well, excuse me. A population here is a meta-ecosystem. So here we got a little bacterial population, let's say. That's a meta-ecosystem. And we have within it autocatalytic cycles. And the cycles that we're focusing on here are the gene cycles. They also often entail other cycles, but let's focus on the gene cycle. If over time, the frequency of that red autocatalytic cycle changes, let's say it increases, we call that evolution. It could be due to chance, just which of these cells survived. It could be due to the fact that that particular gene or that particular cycle somehow results in an organism that is better at surviving and reproducing. That's selection. So that's change that's Darwinian change. Well, you could certainly make a case that you can view ecological succession through the same lens. So ecological succession is a meta-ecosystem of these little local patches. They change over time. Changing over time involves autocatalytic cycles, such as the life cycles of organisms, changing in frequency. They can invade certain sites, they can go extinct in certain sites. And so over time, the frequencies of autocatalytic cycles can change. So there's, again, an equivalence. That's like evolution, but with succession now. Now, general conception will be most of these changes are driven just by the ability of the individual species to survive and persist. But in an environment where there's some regime of disturbance, it's certainly possible that a certain species or set of cooperating species that forms a higher level cycle, they could be favored because they actually make their ecosystem more resistant to disturbance or better at moving into vacant sites. So there's a potential for an analog of selection, even in succession. And this is something that ecologists have discussed on and off. So I think there's some good equivalences here. So the basic hypothesis then is, to come back to it, is that if we can claim that Darwinian evolution and succession are continuous, they're both different kinds of ACE processes, autocatalytic chemical ecosystem processes, then the hypothesis we're really advancing is that something like succession happened spontaneously before there were organisms. And that then the features that we associate with Darwinian evolution, they arose over time. And so the next thing I want to try and convince you is that even without organisms, there would be ACEs, there would be autocatalytic chemical ecosystems. So the first thing I need to show you is that in the kinds of spontaneous chemistry that happens without life, there are autocatalytic cycles, and there are. There's some very famous ones, like the four-mode cycle. This is a cycle that can feed on formaldehyde, and once you've got at least one molecule of one of these little sugars, it can just spin out and make, it's actually a more complex cycle. You can actually make some big sugars as well. And it's known to be autocatalytic. It's easy to study in the lab. And we've done it in my lab even. So that's a well-known one. There are also, though, if you take networks of chemical reactions that have been determined. So for example, Jen took Zach Adam in the bacteriology department had created a database of radiolytic reactions. These are totally non-biological reactions. If you look in that database, there are uncountably many autocatalytic cycles, including this one. This one feeds on visible light, NO, and methane, and generates some waste. But it's an autocatalytic cycle. So prebiotic, before life, there were cycles. Those cycles had ecology. Now, I didn't mention this before, but one of the kinds of interesting kinds of ecology that underlie succession are ordering effects, dependencies, trophic hierarchies. So just to kind of give you the concept here, imagine this little autocatalytic cycle. It feeds on X, different kinds of food, denoted X. And every time you go around the cycle, you make 1A, goes to 1B, goes to 1C, goes to 1D, goes to 2A's. So you're going to have 2B's and so on. So this as you go around this cycle, the number will increase. It's generating a waste product. This waste product might be needed for this second cycle. Because of this particular relationship, the yellow cycle cannot establish, even if you introduce an H, nothing will happen unless you've already got the green cycle active. So there is a trophic hierarchy. And it turns out these trophic hierarchies exist in chemical reaction networks. And just to give you one quick example of this, in that radiolytic network that Jen analyzed, I've showed you this autocatalytic cycle here. If you introduce basically almost any of these red species, you can trigger this, not any of them, but many of them, you can trigger this cycle. And then this cycle produces formaldehyde. So once this cycle is triggered, but not before it's triggered, the full mode reaction can now be activated. So there's like a trophic hierarchy. So what that means is that these prebiotic chemical ecosystems, you could get these cycles. Some of them would be seeded spontaneously. They would start going. Once those cycles are active, they are producing more chemical diversity that allows other cycles that would not previously have been viable to be triggered and established, to be seeded. And that seeding event where one molecule, just little as one molecule comes in, starts reacting and then closes the cycle to make more of itself and amplify. That seeding can happen through multiple ways. It could be some sort of import of a chemical from the outside. So if you've got some complex chemicals that are produced in space, sitting on a comet, and that comet plummets into Earth, you're going to introduce some chemicals that could trigger new autocatalytic cycles. It could also happen just on Earth, but somewhere else, some other environment where the chemistry is a bit different, and so different things are being produced. One of them can come in, that chemical can react with what's there already, and can start amplifying its own production. It can also happen, and this is important to remember, through rare chemical reactions. We always like to think of chemistry as it's just so fast and so predictable. But in fact, if you think about the immensity of chemistry, there have to be reactions that happen only once in a blue moon. And so those reactions can then have the potential, every time they happen, maybe they produce a molecule that can seed a new autocatalytic cycle, generating sort of a new state for that ecosystem. So the vision then that we have is that before there was anything we would call life, there would be these occasional seeding events where autocatalytic systems were seeded. And just playing with it, imagine that there are some subsets of autocatalytic cycles whose members happen to stick to this kind of rock. Well, then you could imagine that different patches of rock, depending on exactly which seeds arrive when, you could have different ecosystems start to arise. But then, of course, the potential for movements between these patches generates a dynamic at the meta-ecosystem level that can look very much like evolution. That's the concept. So I've made a lot of claims. Feel like I should provide data before I go on. And I'm not going to provide a ton of data just because it's turning out not to be so easy. So this way of thinking has led my lab now for the last 6 years or so to really be trying to ask the question, if you take real chemistry, the kind of chemistry that would have been around on the early Earth, is there evidence of these kinds of dynamics? Can we see things changing over time that look like succession? Can we see effects of seeding? That's to say where, you introduce a little bit of something at some point in time, and it kicks that ecosystem into a new state that it can maintain from then on. We call it chemical memory. And can you see historical contingency? So in other words, if you had multiple lineages, multiple meta ecosystems, and they were left on their own, could chance come in such that over time they would sort of diverge from each other, which is the characteristic we associate with evolution. And the basic way we study this is a very simple-minded class of experiments that we sometimes call chemical ecosystem selection experiments. But basically what we do is create microcosms that we want to study with some kind of food. And that food is going to be replenished periodically. So what will happen is we'll make a vial of some kind. It could be a well in a plate. It might have often we include things like minerals to provide sort of structure to it, but not always. We incubate it for a time and then transfer some fraction of that into a new vial and replenish it with fresh food. So in the natural environment, you have fluxes constantly coming in of new food, new energy. We're trying to mimic that by periodically replenishing with dilution. And then we track these over periods of time to look for changes. So one idea is we can take them in the same environment and we track the lineages over time, and we might expect them to become more and more different over time as they accumulate these chance events. That would be a beautiful result, as we haven't seen. Another possibility might be, like I said, we might introduce some chemicals at one point in time, and then observe that the lineages that had at one point in time been seeded by these chemicals remain different even after the seed molecules themselves that you originally introduced have been diluted away to non-existence because of this dilution of regeneration. So those are the kinds of experiments we've been doing, and it's complicated, turned out to be complicated. Analysis is complicated. We're getting better at it. And I'm not going to give you detailed results. My student Tima is going to give a talk later in the semester. He may talk about this work. He may talk about something else. But I'll just to give you a sense that we have several times seen kind of non-linear behavior over time. So in an early experiment that Lena Vincent did, we found this sort of what looked like boom bust cycles, which can happen in ecology, but we don't really know what's happening here. But we haven't seen sort of steady change over time indefinitely, which is what we would love, which would look more like succession. We also haven't yet seen evidence of historical contingency. So here, this is the 10 generations, 10 transferred. But if there were historical contingency, if you took 10 lineages and started them at generation 0, by generation 10, they might be quite different from each other just because of accumulated chance events. Whereas controls that are restarted every generation should have less variation. But no, the amount of variation doesn't increase over generations. And this, by the way, is looking at total chemical fingerprint using liquid chromatography mass spectrometry. Seeding, there are sometimes we see unexpected differences between seeded and unseeded lineages, but not consistently. It comes and it goes. And I now believe that we're going to keep doing these experiments. We're going to get better at them. But we have a challenge, which is in the lab, in a biologically full world, there's just so many chemicals that are getting into our experiment. You can't keep everything out. You're going to have single molecules of everything in every vial, basically. And so we think some of the problems we're facing are because we're on such a chemically complex world. And then if we could figure out ways to do this in a much chemically simpler world, we might see these dynamics. So that was just to say, with a lot of what I do is in the lab, a lot of what we do is in the lab, and we're still working at it, but we do not have any smoking guns to share. So I'm going to pretend that we do, though, and say, let's just, I think we've done enough to show that at least some of these processes have to happen at some scale. We just may not be able to find the right conditions to demonstrate it in the context of a traditional lab experiment. So let's just assume that this is happening, okay? Where does this lead us? So what this leads us is to this idea that maybe it's plausible that this sort of ecosystem succession-like process spontaneously was triggered and then gradually became more like Darwinian. So what I haven't yet discussed is how succession, like dynamics, differs from Darwinian evolution, because I'm sure you all have this intuition. They seem kind of different. So that's what I want to do now for the rest of the talk. So let's contrast conventional Darwinian evolution with conventional ecological succession. So these little diagrams show this. On the left, we have a cellular population. The cells are dividing. The gene life cycles, those particular autocatalytic cycles are represented by the colored dots. They get passed on. Every so often, there might be a mutation where one is changed, the red one becomes the yellow one, and then the yellow one gets passed on. And in contrast, here we have a little messy ecosystem with 3 ecosystem patches. 2 of them occupied at the start. You know, one of them gets cleared out by some disturbance event. One of the ones that was clear gets colonized mainly from this one here, from the middle. And we get a dispersal event from the outside somewhere in here, like here's one that came in from somewhere else, from species. And over time, the composition of this messy ecosystem changes in the same way that the composition of this messy ecosystem can change over time. This time slices we're looking at down here. Okay, so what are the obvious differences? Well, the first obvious difference is that the biological ecosystem, they make their own boundaries. And the term that people use for that is they're autopoietic. And whereas, of course, most ecosystems in nature that we think of, the environment sets a boundary. Or maybe there is no sharp boundary, there's just continuous space and separation by distance. So that is important, that difference is important for really one reason, I think. And that reason is that because of the way cells work, where they create their own boundaries, they grow, they divide, and that generates a high probability of co-dispersal of the autocatalytic cycles within each of the cells. So these genes are passed on as a group to the daughter cells. They co-disperse, they go together. And that's important because the interests of all those autocatalytic cycles are aligned. They are all invested in the same ecosystem success. Of course, it's not totally true. We know about horizontal gene transfer. We know about things like that. But it's largely that's what happens in biology. Whereas in these guys, dispersal is more independent. Yeah. So let's say in this case, the teal one migrated over here. That doesn't mean that the other ones also migrate to this ecosystem. Different, ecosystems can get seed species from many different places. The other big difference is about this seeding process. So in the biological case, the seeding is a rare chemical reaction that generates a new chemical, namely the mutant sequence. In the process, it kind of gets rid of it probably removes all copies of a previous kind of chemical, the ancestral sequence, ancestral allele. In the seeding is a big difference in traditional ecology, in that usually in traditional ecology, the seeding is an import from a regional species pool. Now, the reason that matters is that in the biological case, the new seeded chemical and the site is one that is very similar to one that was already there. And it has a high probability of making a viable cycle. And that viable cycle is going to be probably, all things being equal, quite similar to the one that was there before. It's a tweak. And the mutations generate tweaks and allow for efficient searching of adaptive landscapes. In the case of seeding from import, there's no correlation between the species that is imported and what was there already. And so it results in a much less efficient search process. Adaptation should be less efficient. Now, this distinction is not as clean as it may sound, because in the case of a prebiotic chemical ecosystem, some of the seeding events will be rare reactions, and the rare reactions are going to take something that was there and modify it. So there will be tweaks. So it's a gray distinction. It's not sharp, but this is a distinction of degree that's worth highlighting. So the question this should raise is, how do we go from something that looked like ecological succession in chemical ecosystems to Darwinian evolution? And I'm going to break this down and say, let's think about the two things separately. Let's first of all think about autopoiesis, the co-dispersal of. So we have been we started studying this, and to study this, what we can do is we can simulate the behavior of autocatalytic cycles in some spatial context, but in spatial context. So here we have a meta-ecosystem with hundreds of little ecosystems arranged in a hexagonal array. The green on the right is food. We just introduce a seedling in the middle, one member of an autocatalytic cycle, and that autocatalytic cycle spreads through all those ecosystems and takes over. Now, once we have that machinery, we can then, and this is stochastic modeling, we can then study some of the factors that influence the success of autocatalytic cycles. And it turns out that dispersal ability, the ability to diffuse through the landscape, is quite important. And I'm not going to go through all the details. We have a preprint up about this. But basically, one of the things that's interesting is that if you let's look at study one cycle. If you set up two cycles that are mutually inhibitory, the patterns are very sensitive to the diffusion rate. And what's interesting is that intermediate diffusion rates are the ones that give you the most interesting pattern. If you have very high diffusion, then very quickly one or the other of the two cycles, red and blue, one of them will take over. In this case, blue took over. If you have very low dispersal among these ecosystem patches, then you end up with basically everyone gets stuck, either red or blue, but it's random. But at intermediate values, you get these kind of patch-like patches and dynamic behavior that is distinctive, telling us that dispersal is an important attribute of autocatalytic cycles in general. Now, it turns out, well, this raises the question, well, it's important, but how important is it? So in this set of experiments, what Alex did was take two cycles that are mutually inhibitory. One of them is, he calls it fiercer. It's got more, it's got a higher rate confidence, so it tends to win given they both start off at the same condition. The other one is weaker, but it's faster. It can diffuse quicker. And it turns out that a fast cycle can beat a fierce cycle in some conditions. So there are parts of this parameter space where the faster cycle wins, even though it's always less competitive in an even contest with the other cycle that it's competing against. So we've got this far. We can have some machinery now for studying dispersal. The next step is to really ask the question whether we can get selection for co-dispersal. So the origin of cell-like things, of autopoiesis, is really the origin of sets of cycles that move together. So imagine we had this scenario of this meta-ecosystem with little minimal patches that might be eroding away and being exposed. If at some point some autocatalytic cycle arose that had some ability to get around, maybe it tended to bleb itself into a little blob and grab whatever chemical around it and float around a little bit until it finds a new patch. It would, if it brings with it cooperating cycles that generate the food it needs or generate other ecosystem services, then that new autocatalytic cycle can be favored. And as a result of that, you could imagine over time evolving these sets of autocatalytic cycles whose members move together. And that could be viewed as being the precursor of a cell. So, so that's the hypothesis we want to kind of go on to test now using these models to see if indeed ecosystem dynamics or meta-ecosystem dynamics, when can they favor the origin of these co-dispersing cycles that can be precursors of autopoietic cells. Okay, so I've made the case so far that adaptive evolution and the Darwinian sense it's got a lot of similarities to ecological succession, and that ecological succession would have got started something like that before there was any life, and that process, there are good reasons to think that it could readily generate units like cells, where the co-dispersal or growth division life cycle. What about the next thing? What about the origin of genetic systems? Well, so genetic systems, there's a tendency, I think, to let's call it oversimplify, to think about genetic systems as though it's just one thing. But of course, a genetic system is not one thing. It is a complex ecology of interacting polymers that all do different things. The whole system works because different molecules are doing different things. So instead of asking the question, how did one particular function arise, we should really be asking the question, suppose we have ecosystems, and maybe we have autopoietic ecosystems. Under what conditions could we expect that a polymer that does something useful would actually be enriched relative to other polymers that might be the same length and the same composition? So, so we started to think about that as well. And I'm just going to give you sort of the conceptual and some very preliminary theoretical results. But conceptually, this is the way to think about it, the way I think about it. Imagine you have a basic metabolism. So you've got food, you've got stuff flowing in, and it's reacting in equal autocatalytic cycles, what we can think of as being metabolism. Now, some of those members of those cycles have the capacity to stick together to make polymers. And it turns out that is true of what we know about our metabolism. You know, the central part of our metabolism might be the TCA cycle, and it connects to a lot of building blocks. You know, out of the TCA cycle come the monomers of you know, of RNA and DNA, for example. Well, particularly peptides, I guess. So these guys are just being formed. They're in the environment and they're going to react, and they're going to, whether or not you want it, they will make some polymers of various lengths and various sequences. Now, but imagine that, let's say you get out to 8-mers, and one of the 256 possible 8-mers is a useful sequence. Let's suppose it catalyzes one of these reactions. So it actually it benefits the whole ecosystem because it generates more flux of food into biomass. What would happen? Well, it turns out that if that just happened, there is nothing there's no reason why that particular 8-mer would be more frequent than the other ones. In order for it to be more frequent, you need two conditions. The first condition is you need some way in which selection acts at the ecosystem level. So you need to have a meta-ecosystem where some local ecosystems are productive and some are less productive. Of course, the productive ones are going to be the ones that happen to have that sequence. So that's the first thing you need. You need some kind of selection at the ecosystem level. It's really like group selection. The second thing you need is it could be very, very weak, but you need some positive feedback. If an ecosystem has a particular polymer, it needs to have a tendency to make more of that polymer than another polymer. And in modern genetics, that's done by template-mediated replication. So you need something like that. But if you have both those things, you can just show by simulation, you can get the enrichment of polymers that confer ecosystem functions. So we now have an ecosystem that is starting to have things that look a little genetic. They have some innate tendency to template, and they do useful things. They make catalysts that benefit the whole ecosystem. But that's the beginning. We want to be able to build a whole rich ecological community of many, many polymers that work together. And they have to do a lot of things, including make the genetic system itself work better. So the way that we can think about that is imagining there's another kind of sequence. And I'm calling it a polymerase here, but whatever, a sequence, a different one, whose effect is not on the metabolism, but what it does is it somehow improves the efficiency of that templating process. The templating process is a process that helps make this polymerization cascade tend to concentrate on whatever sequences are there to start with. And so if that arose, then you can, and if it happened to be in a particular community that had the metabolic catalyst, well, those two polymers are there, and they're going to be produced at higher rate when that polymerase is there than when the polymerase isn't there. So it can also be favored. And we have some very preliminary analyses that kind of demonstrate this effect. This is actually, we call them different names. We call them a ligase, not a polymerase, and kinase is the metabolic one. But the basic idea here is that you can bootstrap longer and longer polymers that confer diverse benefits to the ecosystem. And some of those benefits are improving the efficiency with which template-mediated replication happens. In other words, the genetic system itself can evolve by progressively addition to this feedback, these feedback mechanisms. So what that leads to, and we obviously need to do more of this, but what that leads to is this kind of concept that over time, the ecology of the cell could accumulate and become more and more familiarly genetic. I don't think there's a moment in time when it undergoes some kind of quantum leap from one kind of evolution to another kind of evolution, from succession to Darwinian evolution, for example. But we can say that over time, it becomes, inheritance becomes more and more reliable, evolution becomes more and more efficient, and it becomes, in some sense, a self-accelerating process where evolution itself evolves. So that's a kind of overview of what we've been doing. I will just add one little last piece just to say that I'm very lucky to have Praful. He is a recently graduated physics student who worked in my lab for the last couple of years, collaborating with Eric Smith there on the right. He's been doing some really beautiful mathematical analyses of chemical networks and their kind of behaviors. And what they've been able to show already is some really interesting sort of implications that if you sort of generalize chemistry, you can find cases in which, just like in biology, you can find chemical networks have the ability to progressively tend to get more and more out of equilibrium with the flux of monomers. This is imagining you've got a polymerization system, monomers coming in some way, and you could have average polymer lengths kind of keep increasing over time in this rather stepwise stochastic process. Moreover, this process can show historical contingency where if you start off at that star, you either go to that blue, you either go over that blue settle point to this star, or you go over that blue point to that star, and then you're on a different trajectory. So I think that's maybe this is maybe telling us is really what I think might be the thing that I've come to believe more and more, which is that when we look at the origin of life, we have a tendency to be just awed, like Darwin, by the complexity of what we see on the planet, and think it must have required some really special things that happened and were very unique and quirky. But I guess I'm going to suggest that we don't know that a case can be made that actually the emergence of life is a gradual transition. It's not like one moment when it comes. It's the gradual morphing of an evolutionary process, that this process may have some predictable features. For example, it may tend to constantly evolve, using energetic resources more efficiently at least biosphere level. And that as a result of this, it may be much easier and more common than we assume. So personally, I'm convinced that even in our own solar system, there are other systems that will have gone some way down this road. And I remain optimistic that we can at least see some of these kind of dynamics getting started in the lab. We haven't done it yet, but I don't see any reason why it won't be possible, in our lifetimes. So sort of just to kind of give you this sort of perspective then that I have acquired over these last few years, I used to think of life as particular. I would look around this room and I would see however many individual lives there are in this room. And I think we have a tendency to do that. I've come around to thinking of life as one whole system. It's a planet-wide system where a long time ago, about 4 billion years ago, some autocatalytic chemical ecosystems sprung into existence. In protoplasm, if you will. It had this ability to make more of its components, and it made more of its components and spread. And in spreading, different pieces of it became different. And it ultimately covered the entire planet with many, many variants of this original protoplasm that are just all continuous through time with one another. That an ancestral protoplasm is in all of us, and it's in everything that we call alive, and it's totally reshaped the planet. And then I don't think we should think of it so much as a set of individual things. I don't think we should even think of it as distinct from the planet. I think we should really think of the biosphere as a system that has got probably some very, the laws of physics are basically acting to basically more efficiently use energy that is constantly flowing onto the earth, and that we're sort of dissipating that energy. We are part of the earth, and we are part of the way that the earth is dealing with the influx of energy, sort of in this predictable way. And so when I go back to Darwin's quote, I wanted to do this at the end to point out what I think is, I mean, he's a brilliant guy, right? We're very intuitive. Is the second thing we often overlook, which is, I think we often focus on the elaborately constructed forms, the individual things that blow our minds with their sophistication. But really what's cool about this is this interdependence of all the pieces. That within ecosystems, meta-ecosystems, within the biosphere, at some level, everything has to work together, and it does. And I think that when we take that perspective, it's much easier to understand how a system could get going with that interdependency right from the beginning, all these different autocatalytic cycles codepending, and then over time complexify, diversify, but with always this sense of interdependence among the parts. So that's my sort of grand philosophical send off. Just to end, I want to thank. So much of the work you heard about is a recent NSF grant that I have with Emily Dolphin at MSU. She's a computer scientist and artificial lifer, and Eric Smith, who's a physicist. And I've had wonderful, diverse students in my lab over time, and a bunch of really great collaborators at WID and elsewhere. So I guess with that, I'm happy to see if you have any questions.
Right. So, what do you think the implications of your work are for the debates about protein first versus RNA world system? It seems to argue very much against, at least implicitly, an RNA world, and that heredity is kind of added on afterwards to stabilize what's already there. Is that an accurate interpretation?
So I'll repeat the question for the people online, yeah? So the question was, there's been a bit of a debate in the origin life field about whether it's RNA first, protein first, metabolism first, what came first? And the question is, well, what does my view say about this? I guess my view would say that it's probably all of the above. That I view it as likely that at least short peptides, short nucleic acids were all around very early, as with metabolites. One of the big questions that I don't know the answer to is, at least the tightest feedback loop with the polymer is template mediated processing, which we know happens well with nucleic acids. There are some chemists who've shown similar phenomena with peptides, although less efficient. I don't know whether nucleic acids will maybe, because of that property of nucleic acids, whether they played an outsized role in this transition to genetics part of it. Did I answer that?
Yeah.
Does that make sense? Yeah. Yeah.
So I usually think of adaptation as being often a response to limitation. And in your models, you're typically assuming sort of an infinite influx of things. How would that perspective shift? Would it shift? That's great.
Yeah. So the question was, well, I mean, adaptation usually implies some kind of limits on what's coming into a system. And in fact, physically, we know all systems, they don't have infinite resources. And it sounded like my model was implying sort of an infinite flux. Actually, we don't assume an infinite flux. We just assume a constant flux. Like we assume that, there's a certain rate at which food enters the system. And so the result of that, the dynamics we expect to see is not an exponential rise to infinity, but a logistic growth and then stabilization. So, and yeah, we see those logistic dynamics for chemical cycles as you do for ecology. So, but yes, good point. Yeah.
Curious also about what your models say about just how quickly we think life's growing in our world versus other ones, and whether like the ideas of seeding from whether it's a comet or something else is a necessary component in order to get the kinds of larger molecules that might have been able to spur it so quickly. But presumably in your simulations that you're going to ask whether or not over like time this is necessary to, like in the time we saw life spring up, you think here. But,
yeah. So the question was about the time frame, how long it would take under this way of thinking for life to complexify, and could we figure out with models whether it's plausible? I don't actually know. I don't think we have an easy way to answer that, for the simple reason that chemistry is unimaginably complex. I mean, people, we learn in chemistry there are these kind of classes of compounds. We learned that in biochemistry there are this number of metabolites. But in fact, at small abundances, there are huge, I mean, millions of different compounds that are present that arrive. Like the Miller-Urey spark discharge experiments, where you put lightning through gas mixtures. Yeah, you get amino acids, and you get lots of other things. People have estimated that there's hundreds of thousands of compounds in there at low concentration. So I'm saying that because it makes it a little hard to know how rare and how critical individual seeding events would be in this scenario. And we also don't know how to calculate those rates. So I think we're in sort of a realm of, intuitively, it feels like if you have little local patches, these kind of dynamics can happen, but we don't really know how long you would have to wait on average for a key chemical to seed the next tier in the trophic hierarchy. So the model is, I don't know. I mean, I have no reason to think that it's impossible. In fact, the fact of doing experiments in the lab implies that I think it's likely, because otherwise we wouldn't be doing them. But I have no way to be sure of that. And if you can think of a way to calculate that, it'd be great. So, yeah, how long would you expect it to take?
All right, well, we are out of time, so please, big thank you to Professor Lynn.